would like to introduce the first speaker, Irene, who is working at the Department of Physics at the University of Hamburg, and specifically with nanostructure and solid-state physics. And Irene is also international experience and has experience as co-founder of a company in San Francisco. So please enjoy your presentation, remember to write questions, and welcome to Irene.
Okay, thank you for the intro. Let me share the screen. So yeah, I made this presentation a bit different to what I'm used to present. So there are no scientific results here. It's more about me and my experience with industry, both from the scientific point of view, so working with industry as a researcher, but also working with industry as a startup founder. So I will just give you some hints of my experience, what I think worked, what I think were the main challenges. And then, of course, I'm open for discussion. And of course, if you want to know what we do in the lab, I'm also happy to discuss at some other point. So as Stefan said, I'm a group leader in the University of Hamburg in the physics department. And the main work of my group is using nanotechnology for biomedical applications. So we basically make devices that can be used for DNA analysis or for protein analysis. But before I made it to Hamburg, I traveled around the world. So I'm originally from Spain. I did my PhD in Barcelona. And the main focus of my PhD thesis was on nanofabrication. And I will tell you some background on that in a minute. Then I moved to Copenhagen as a postdoc, where I spent a bit more than a year working on using this. So first I developed devices and fabrication methods to make these devices. Then I started using them to understand some fundamental properties of light. So confining near field and plasmons in big groups. And then I moved to Berkeley with Marie Curie. And then that's what for me was an eye-opening experience. I spent 4 years there. And the fact that I was surrounded by startups and industries and being at the core of the Silicon Valley and people, industrial collaborators and colleagues going to industry or new colleagues coming from industry, that was really a big experience for me. And I really enjoyed that part where you do something in the lab, but you always keep in mind who could benefit from this. So then I really started using my devices for things that could be applied. And then at some point I started a company, but I will also tell you some details. And then at some point I decided to come back to Europe. So that's where I'm now. I'm a group leader in the University of Hamburg. But I think now I'm combining all this expertise in nanotechnology, devices, and especially keeping the industry or the innovation in mind. And I think that's what I'm trying to do now in my group. So I always had collaborations with industry all along my career. And I think it really started during my PhD thesis. So I was part of a really big European project, the NAPA project. So this was back in the days when nanoscience and nanotechnology was starting. And then all the electronics and all the chips need to be made smaller and smaller and smaller. And then we had to develop new tools to make these chips smaller and smaller. So I was working for this NAPA project. So that's nano-patterning at the nanoscale. And then it was a really big project. So I think we were 35 partners. And there were universities, research institutes, but also a lot of industrial collaborators. And they were coming from all the different points. So there were companies making materials, so polymers or metals. And there were also companies making tools, so large tools that you can make to make nano fabrication. But there were also companies doing applications. So I remember we had Fiat. So we always had industry involved in all the levels because you want to keep them in the loop, keep an eye on who could be the final user, and then also keep everything, something that could be compatible with mass production, something that could have a utility at the end. Then along my career, I kept some of these contacts, like microresist technology, but I also made new ones, so microliquid or Carl-Zeiss. And what I think is that the collaboration with industry is definitely a win-win situation. So for the companies, they get visibility because you present and you give their names in companies. So you are a user and you make advertisement for them. You show the potential of their products, especially when they are used for applied research. So for example, I'm making devices that could be applied for, I don't know, for detecting cancer biomarkers, and I'm using a specific polymer. So this company making polymers now suddenly has a foot in the biomedical industry. So companies, they also stay updated with the state-of-the-art research activities, so they know what we are doing, and they might even get new ideas or applications for their products. And then on the other hand, we get to try new products. So for example, these polymers, in the lab, we have a variety of polymers which are not commercial, or we also can use demo tools from a microscopy company. So we get access to tools before other people can buy them. We also get to work with a specialist. So when you have this kind of collaboration, you can take the phone and you can call the guy who made the software for this microscope, and you can talk to them, and you really have a very deep understanding on the materials and the products that you are using. And also you see a very different way of working. So I think when you collaborate with industry, you see how they are very goal-oriented and how they structure their day and their work. And I think that that's something that could be useful also for researchers. And also for us, it's always good to keep an eye on what society or the market will need. So you develop something, but at the same time, you develop something that at the end of the day could be used, I don't know, for detecting toxins in fresh water. And for me, as mentioned, networking was crucial for me, starting from my contacts or your contacts. You will always find somebody who is interested in your work. You can also find industrial partners in conferences, because then I think there is already a filter. So the companies that go to a conference is because they want to be involved in research. So then the first barrier, that it's people who don't care about research, it's gone if you go to a conference and you talk to them there. And then also involve them in the project. So from the very early stage, when you are preparing the proposal, you should involve them, because then you have the commitment and you have their interest. And I also saw that exchange of people also works. So when you go to the company and you do something with them, or they send somebody to you, or you have colleagues who work in a company, and then you can exchange ideas. So that's what I found that was very effective. Challenges is that, of course, we have different standards. So something that for you looks good might not be good enough for them. So we speak a different language. We also like to give details. So we really like to say, we do this way, and then the industry doesn't. So this can be a problem, for example, when you want to write a paper. And then I think you have to find the right person and the right company, because not everyone is interested in doing research. So, and then also, as mentioned, I also started a company in Berkeley, and then we are also trying to start a company in Hamburg. So in Berkeley, we wanted to make nano devices for detecting cyanotoxins in fresh water. And now we are trying to make nano devices for liquid biopsy, so for biomedical application. And then I think when you try to collaborate with industry, when you are a startup, then the rules of the game change a bit. So then here, I think industrial partners are really key. They are essential. I mean, when you are a researcher, it's a plus, but when you are a startup, you really need them, because they have experience in upscaling products, in developing products, in throughput, and we really don't have that experience. So I think you really need a partner, but you need stakeholders, you don't need competitors. So we have this tendency that if you are doing microscopy, you contact microscopy companies, and then suddenly you are competing. But instead of that, you have to see who are your key collaborators. So somebody that can help you with something that you don't know how to do. And then, of course, you should contact them when you have a working prototype, because then if you are at the very early stage, you think it's a product because you had a measurement, but then probably for them, that's not the case. And then they might lose interest in your stuff because they will believe it will never work. So as a conclusion, we need them and they need us. For me, I think it's really crucial to find the right person and the right company, because we also had a lot of bad experiences with companies or people who did not care about research. Or if you also approach them and you are in too early stage, they will think that we are just playing with things in the lab. And one of my main advice will be to involve them in the project. So from the very early stage, because I think there you already filter and you already have people interested, and you will also have the commitment and you will focus the whole work in the common interest from both sides. So that's it from my side.
Thank you very much, Irene, for also keeping on the time schedule. I really like that you presented both sides of the story, and I appreciate your experiences there. I don't think there's at the moment any questions to you in the chat, so I think we'll just go on to the next speaker. And then, of course, a kind encouragement to you, if you have questions, please write them in the chat. The next speaker will be Ute Krengel from the Structural Biology Group at the Department of Chemistry at the University of Oslo. Ute has also an international background and has been involved in, I think, three of the Halo 6 months project, or at least being the main applicant on that. So I look forward to your presentation, Ute, and please go ahead.
Thank you very much, Stefan. Yes, so this was an interesting invitation and new for me. So like Irene also mentioned, I never gave such a talk before, and it made me sit down with Katja's questions and think about what experience do I really have. And it turned out I felt that I had more experience than I had originally thought. So I was thinking, so I'm a structural biologist. That means I'm interested in protein structure and function, and in particular protein carbohydrate interactions and enzymes, so a completely different field compared to Irene. I was thinking to tell you a little bit more about my career and then the industry contacts throughout that. So first, I studied chemistry at the University of Göttingen and did my PhD in Heidelberg. And after that, I had my first postdoc at the University of Toronto in Canada. But the first industry contacts were really when I went to Groningen, where, by the way, I also met Marjolein, who I see is on the audience, so that's nice. So this is, there I was hired on a project that was from industry. So I had to solve the structure of the xylanase in the together with the company Gist Procades. And they were using the xylanase as a supplement in chicken food. So usually you want to maybe think of getting thinner in the common world, but for them, it was, of course, more interesting to get a big fat chicken with just adding a little bit of the enzyme, and then they could make much better use of the food, the polysaccharides that they were eating. And in addition, it was less, it was more solid waste also, so easier to clean up. So their interest was, first of all, how would this enzyme function, but then also they were interested later on to maybe use other applications, for example, for making bio-bleaching agents, so that you can take less chlorine, and so you have to work with an enzyme that can work at high temperatures. So if we had this one xylanase here, maybe we can use protein engineering when we know the structure and function and the enzymatic mechanism to also use it for other purposes. So here I was actually not approaching industry, but I was hired on a project where industry was interested in finding out more, and they contacted my boss, Bauke Dijkstra, and then I was hired on the project. And I mean, that is something that still once in a while happens today, that companies contact us because they are interested in getting a structure solved. So it's not always such that we are the ones contacting, but we are also contacted. And in the sense, works fine because I'm leading a core facility in Oslo, and I'm part of a national Norwegian core facility. So we actually benefit from getting some money in and providing some services. Another project also in the Netherlands, in Groningen, was on antistasin, which is a protease inhibitor, so for inhibiting blood clotting and so on. And there was a collaborator on this. I think he was not in industry when he started out with this project, although I'm not 100% sure, Hermann Schröder, but he worked at Höchst at the time and now in Sanofi and still very much interested in protease inhibitors. Then I moved to Sweden. So that was a 7-year period there where I had several industry contacts. For one, the first, my husband worked at Astra, so obviously we had some common friends there. That was before it was AstraZeneca. And actually the reason why we went to Sweden in the first place was that I applied for a position in industry, which I didn't get. So when there was a second position that potentially my husband could get, I urged him to apply. He was actually hired before me. And when I was at the interview, they told me he would be hired, but they didn't realize that he was my husband because we had different last names. So I said, you better hire me too, but they didn't. But so I got into academia more as a original second choice, but I very much enjoyed now. So I have always thought I would end up in industry, and here I am at a professor at the University of Oslo. What was a bit difficult for me in the beginning in Sweden was that I already had 8 years since my PhD, and essentially then you are out of the business. You cannot climb up the ladder. So I was very lucky that there was, from this Swedish Strategic Foundation, there was a position for a forskare, which didn't need the forskarassistent medium position. And you can imagine I applied very well for that interview. And if I would have known who were my competitors who were already professors, I would not have thought I had any chance, but I did get it. So I was still there. And that was from the GLIPS network, glycoconjugates and biological systems. And I was also very strongly involved as a participant in the Swedish structural biology network. Those were the two most successful of these networks, and SBNet is still going strong. And I hope to be joining to Taberg in June this year. So these are graduate networks. They finance PhD students. And that was actually a great way also to get in contact with industry. So, I mean, if you, both in the terms of, SBNet, for example, they're also the industry representatives, they are there. It's usually small small conferences, so you get to meet anyone you would like to meet. And when you know someone already from previous experiences, then it's when you need them, it's much easier to use these contacts. And I was in the board or associated to the board for GLIPS, so there I had a more prominent role. Also, in Scandinavia, many of you might know, it's not always so easy to get contact with others, but I met a PhD student from Cuba. He was just working under us. Of course, two foreigners meeting each other, we got to talk, and he worked on anti-tumor antibodies. There's a whole biotechnology institute where it goes from research to all the way to the clinics. And so we started a project together, and that is still working, although he moved to Colombia. So this is one of these industry projects which was really going all the way, and several of the anti-tumor antibodies are on the market, and we help with validation and structure functional analysis. Now I'm working with local colleagues also on Nextera, And there are 2 more graduate networks, Biostruct and BioCut, and the Norwegian core facilities, Nordkrist. I've coordinated some ITN proposals where then there was a whole different need of contacting some industrial partners, and I have some other industrial collaborators. How much time do I have left? Can you tell me?
A few minutes. 2.
Okay. So, I mean, with the ITN coordination, I think it's actually interesting because there I didn't know all the people already who I contacted, but I had to find the best partners. And then either I went through colleagues who knew them, and that was also non-governmental organizations who were important there. Or I wrote like a 2-page description of what I wanted to do, and I targeted for the right industry and let them think first, and then talk to them on the phone to see if we had a common interest, and we got all the companies on board that we wanted. So some of these here are direct collaborations, some of them are indirect collaborations, and then some contacts. And of course, I have a lot of students who ended up in industry, and so there are also these kind of contacts. And it's actually cool if some companies are asking, do you have another good student? We have a position to fill. So I might not have a lot of time for Katja's questions now. How do you communicate your life science research to industry, and how do you engage companies in your research? But I think maybe I've already answered that a bit, or else I'm open to questions. So the value of the coffee break, have a joint, I think, Katja, Kajsa, you mentioned in the beginning, it's a pity that there's all these webinars in Zoom. I mean, if you really meet in person, because what I think is really important is that, for one, you have the same interests, but also you have to like each other. If there's no personal chemistry, it will not work very well. So, I mean, to make these collaborations just via Zoom is a hinder. So I'm looking forward to more in-person engagements. I have, for example, with the ITN, I also contacted spin-offs, and there, Peter Seeberger, for example, he was invited as a colleague, and I invited other industry colleagues, for example, for talks, and then you get to meet them, of course, also. So that is another chance. And in the GLIPS network, for example, we let the PhD student invite someone, and that was Peter Seeberger, who then was in my ITN, one of the startups. So there's a lot of possibilities also with, like recently there was one of the BioCut school board members from industry, where we saw that he's starting up companies and trying to get investment money. So this, we liked each other. So there we thought we can do something here together. Yeah, I mean, if you have the same goals, then this can be very good. And life science research, I have a good friend who is working at Abbott in the US, quite high ranking. And he says, I mean, he is working very hard, but he is head of a big team. And it's just so nice to see that the work he's putting in is benefiting patients. And so this is for the field I'm working in. That's, of course, a great outcome. In another project of mine where I'm working on sustainable energy, it's a completely different type of industry that I have to contact. And finally, you mentioned the 3 HALOS projects. It was completely different HALOS projects. So I, of course, also contacted different industry representatives. So thank you.
Thank you very, very much, Ute. That was insightful. And I also appreciate what you said on also, Irene, that this is a different type of presentations than what you're used to, but I like this presentation, so keep doing it. Before we move on to the next speaker, I had one practical question for you. What I appreciate what you mentioned is that sometimes companies reach out to academia rather than the other way around, which we typically focus on. We typically focus on reaching out from academia to industry, and for good reasons. But I'm glad to hear that it sometimes happens the other way. And can you say a few words about how can you improve that as an academic researcher? How can you improve your chances, so to speak, that companies reach out to you?
Well, I mean, for me, I think it's not that difficult because I'm heading a core facility. So we are having outreach events. Just today, for example, you know that I'm double booked. I had a talk just 2 hours before, and there we presented also our core facilities. So before giving my scientific talk, we told the audience what we can offer to them. And so then the word gets around. So then we often invite speakers to University of Oslo. So when I took a leadership course here, actually everyone noticed my very strange name, which in Scandinavia is very peculiar. And so they knew me from all of those activities. So I think you have to be visible in one way or the other. When they know what you're doing and you're good at it, then if they need your services, they will contact you.
Thank you, being visible. And when you say they need the service, of course, that means they need to know the services that you have. So
yeah,
indeed, the visibility. Thank you very much. And I also just discovered that we actually were at the same university together at the same time. I was a PhD student at the University of Groningen, so maybe we've met before
okay.
At some point.
So I can practice my Dutch with you again.
Dat kunnen we inderdaad doen, yeah. But let's not today.
Yeah.
Thank you very much. Let's move to the third speaker of today. And again, thank you to you, Emmanuel, for joining us, being ready at so short notice. So our next speaker is Emmanuel Larsson, who is at the Division of Solid Mechanics at Lund University, or a technical university, and is also connected to Max IV. And also Emmanuel has an international background, but I also think it's interesting that he will be, I think, also speaking a little bit about other types of studies or materials rather than only life science. So Emmanuel, please go ahead.
Yes, thank you, Stephen, for the introduction. Yes, I can mention that I am at Lund University Division of Solid Mechanics, but I also have affiliations to both LUNAC, the Computing Center, and LINX and SIPA. And I have a connection to Max IV mainly via the 4Max portal, which is a project that we're collaborating on. And I'm also connected to the KIM, Center of Quantification of Imaging Data from Max IV, which is a collaboration between Lund University, Max IV, DTU, and Copenhagen University. And they are also part of the HALOS project. I will start with presenting my background, and you will see how I gradually moved into collaborating with industry. So I'm actually from, I was born and raised in Linköping, and I studied a Master of Science in Engineering Biology, Devices and Materials in Biomedicine/Biophysics at Linköping University. So I'm a little bit of a funny replacement for Maria Sunnerhagen, who is actually a professor at Linköping University. This image here shows you an overview of the Ångström laboratory, which is where they have below ground an electron microscopy laboratory. And I'm showing it mainly because they are, let's say, accelerating in a way electrons or moving them in some way. And it's also a circularly shaped research facility. And you will find out why this is important soon. In 2010, I exchanged let's say the bad luck local landline number for the phone 013 to 4040, and I moved to Trieste in Italy, where I did my master's thesis on, let's say, bone engineering scaffolds, human skull bones, and I later on stayed for a joint PhD together with Linköping University, University of Trieste, but mainly stationed at the Lettra synchrotron, where I looked into, let's say, the application of contrast agents into mice lungs, which were artificially induced with asthma. And so this is the biomaterials background of my career. And then in 2016, I made a postdoc at the nano tomography end station of PO5 at DESY, which is operated by Helmholtz-Zentrum Geestach, which now changed name to Helmholtz-Zentrum Heron. And here we looked into various material science-based applications, which have, let's say, quite a clear connection to industry in a way, but we more did the research on a more of academic point of view, but with industrial applications going from jet turbines to let's say the airplane wings, or to the next material in your tomorrow's phones, let's say. And then in 2018, after being abroad for roughly, let's say, 9 years, I was recruited back by RISE, which stands for Research Institutes of Sweden. And I joined this project, which is called Large Scale Research Infrastructure, which aims to a little bit try and get Swedish industry more involved and run projects and pilot projects utilizing MAX IV or ESS or other facilities around Europe or in the world. And this is where I really started working more with industry, and I did a lot of scannings on food science applications, but also wound care dressings, medical devices, and metals. And we actually lived in Hyllie, which is the last station before entering Denmark. And you can see that the train station is circularly shaped, and they have a small light source in the ceiling as well. And since 2020, I'm actually at Lund University, as I mentioned before, with quite some various affiliations. We live in Arlöv, between Lund and Malmö, where they have a circular shaped school, which might be the place where they will, let's say, educate and generate the next generation of tomography experts. You never know. And you can see in my career, I had 040 as a local landline number for 10 years. But so there's a conspiracy theory in my life around circular shaped research facilities or other buildings. But we're moving to Lund in 2 weeks from now, and we're changing 040 to 046. And I have no idea what's going to happen in our lives, but I hope it will be stable. And with that in mind, I will present to you some industry relevant projects that we did, ranging from pharma to medtech, using X-rays and neutrons. And I can mention that with Vinnova funding, I've been since 2018 involved in 10 different projects with industrial partners, and additionally 3 projects on top of that were involved with trying to educate PhD students into using X-ray and neutron techniques, in this case tomography. So during the last, let's say, could it be 8-7 minutes, let's see, I will present 4 different projects that we did together with various Swedish companies. And yeah, one of them, this one with the BioGaia, is actually one of these educating industrial PhD students. And since let's say ESS is not up and running yet, and MAX IV will have tomography available via Danmarks and Formax beamline at the end of the year, we have during these last 4 years carried out a lot of these pilot projects at other facilities such as DSE and at the Helmholtz Centrum here on beamline, as well as at ILL in Grenoble, LLB in, outside Paris in Saclay. So during this time, I've been involved as a mediator working at RISE, also Lund University, and also with collaborating with Swerim. So for the first project that I will show you, we did this project together with Mölnlycke Healthcare, and they are based in Gothenburg, and they had various more destructive ways of checking how the blood spreads in their wound dressings. And it's very important that the blood is in a fast way transported away from the wound into the wound dressing further away, so that the wound can heal without being inflamed. And we use neutron radiography and tomography plus time, so meaning 4D tomography, to study how liquid is being taken up by this wound dressing. So I will show you some animations here. On the left side, you can see that the liquid, the simulated blood, is pumping into this wound dressing. And on the right side, you can actually see the same thing in 4D after we have segmented the image. I will restart it. So you can see here the liquid front going into the 3D space or in time, so that's 4D. And image analysis work is ongoing, and we have a manuscript coming up. And here you can see that just some highlights that we are trying to separate, let's say, new positions where the liquid is entering to try and say something about how the liquid is spreading into these pores. And then for another project together with Sobi, we used a sample environment again, where we put syringes inside the sample environment where we could vary temperature and pressure. And the reason for this is that we wanted to simulate how what can happen to syringes during transportation, because it is a well-known fact that syringes can actually leak during transportation. And if this happens, the company needs to call back the entire batch of that case where potentially only one syringe leaked. And this costs a lot of money for storage, transportation, and not to mention patient suffering if a drug is not delivered on time. And we did in total 2 industry beam times where we managed to monitor this process. And I will show you some of these results in this video. We made also some ways to create an air channel in order to a little bit emphasize that that air can enter in and create a sub pressure, and this actually pulled down the liquid into the needle. And we used neutrons because neutrons are quite transparent to metal, and if we would have used X-rays, it would be difficult because this metal part of the needle would have scattered and leaded to more artifacts. And we could not really use the original drug or water, so we need to use a contrast agent in order to see it, to detect it. In total we did 2 beam times with Sobi, and the work was also presented at an international conference in Gothenburg by Jonas Fransson from Sobi, and it was actually quite well received by the international companies working with syringes and filling up syringes and so on. And for the second last project, we are working on a PhD project looking into freeze drying of probiotics. And during a set of 3 months, we have been optimizing the size of these probiotics, trying to get them optimal for tomography. And this is a reconstructed slice, and I have then trained Shuaibai into image processing and image analysis using code provided by Kim and working on the LUNAR computing cluster at Lund University. And he also presented that work during a summer school last year. And there is a loop in how we work in this way. And the ultimate goal is to use a sample environment, put it at a synchrotron, and monitor this in 4D over time. And to prepare for such a beam time, we have also made some radiography experiments where we can monitor the freeze drying process and just to make sure that we have enough contrast. And this shows you this in radiography, and this we included in an application that was sent in for beam time roughly 2 weeks ago. And then as the last example, we scanned some metals, 3D printed metals. And the reason I'm showing you this is because metals are actually used also in the medtech industry. If you might be in the need of a bone implant or something, it might many times be made out of metals. And as you might know, it's important also in another step that the metal, let's say, is well integrated with the body, because you will have protein adsorption to the metal. So there's a lot of surface physics, biomaterials interaction and so on. So that's the reason why I have this example, and especially I have this example because it was done at the DSC synchrotron, which is a part of the HALOS region, and we used actually the DSC cluster to carry out the image processing. And I will just show you fast here how a set up image processing, image analysis protocol might look like. There are several steps, several operations we need to perform, but at the end, we can, for example, segment porosities and cracks in these generated and printed 3D metals. And then we can use those regions, quantify their sizes, and let's say write out a very long text file with information about a set of parameters as volume, let's say elongation, direction of these pores and cracks. And we had roughly 30 data sets, 65 gigabytes each roughly. And with this protocol that we applied, it took 41 minutes each. And it was run on the DSC cluster, which is a very strong cluster, and using various Python code. And here is an example of what we can produce. We can then plot all of these parameters against each other, try to find correlations, and we can also color code these parameters, these inclusions that we detect, these features, and we can label them in different colors to, in a better way, visualize to the observer, to the industry, what is actually going on in the materials. And with that in mind, I will just thank you, the listeners, and also collaboration partners, and please feel free also to connect in case. Thank you.